structure. Now, I know that it's the class day before the exam. I know nucleic acids aren't technically coverable material on exam 2, but I will tell you that this lecture, nucleic acid structure, even though I'm a protein chemist, this is probably my favorite lecture of the semester, just because I think the activities are really cool and they do a good job illustrating the point that I'm trying to drive home today, which is what are the interactions that hold DNA and RNA together, and what are their relative importance? Because as you're going to learn, different things may be weighted differently than you might have anticipated. But I'm not going to say anything more about that because I don't want to give away like the punchline of the lecture today. So before we dive in, we'll do some housekeeping. Okay, so again, we have our roles for today. We're talking about nucleic acid structure. We have spring break. When we come back, we're going to do a day on protein signaling. So we're going to skip over lipids. We'll do a little like mini lecture on lipids with signaling because most receptors are contained in lipids. And then after we talk about signaling, we're going to have our paper discussion day. So after the exam, the paper will go up onto the Sakai website. You will have it. Shortly later, I will post some pre-assignment questions, and then we'll do a discussion where you'll work in your new groups to answer questions about the paper, and then you will submit those for a grade after class. So that will be coming at you shortly, but for now, you don't have to worry about that quite yet. I just want you to know it's coming. And then the week after that will be the nucleic acid structure quiz. But for now, again, we need to remember today, nucleic acid structure. Thursday is exam 2. Most people did pretty well on the enzyme quiz. But if you weren't happy with your grade or you want to get that 1 point back, you're more than welcome. I welcome you to submit corrections. And those corrections will be due with the exam on Thursday. If you're confused about why you lost points or you still have no idea how to units from the Lange-Burke plot, I talk about it in a video that I made where I walk you through the key to the enzyme quiz. So if you go onto the supplementary videos folder and you find the enzyme quiz walkthrough video, it's 20 minutes, so it's a longer one because I didn't script it. Sorry, but it should. I try to do a really thorough job of explaining my logic for the answer behind each question. All right. Let's see. Okay, and then again, we have an enzyme help session tomorrow, on Wednesday from 6 to 8:30 in Salem 13. So that's on the ground floor of Salem. So if you walk in at the entrance across from the library, you have to go down on the elevator, and it's in a conference room across from a little kitchen. So again, it's fairly informal. You can come in and come out. The risk of that is that we might talk about the same thing twice. But come as you are able and as you have availability. Yes, Paige. Yes, sure. Oh, yes. Thank you for bringing that up. Yes. You may come in and take the exam. Yes, you may have those extra 15 minutes. You may come in at 9 to take the exam. So we will be taking the exam from 9 to 10:15. No, excuse me, 9 to 10:30. We go from 9:15 to 10:30 in a normal class. We will go 9 to 10:30 for the exam. And I'll be here super early like I always am because I like to have time to mentally prepare myself for class or sitting and watching you take an exam, as class will be for me on Thursday. Because I have to do a better job of making a more thorough key for you, which I've already done. Anyway, okay. All right, so that about covers it for housekeeping notes. I've chatted with some of you about how to prepare for this exam. Again, I will say take a look. So look at the problems we solved in class. Look at the problems from the enzyme quiz. If you're thinking about what kind of skills do I need for the exam. In addition to the enzyme problems, the enzyme quiz is a useful tool for you. Take a look at the practice exam for an idea of the types of questions that will be asked. I always recommend going through and I've told you at the beginning of the semester there's always going to be a compare and contrast question. So one study method I recommend is finding things that are similar but also slightly different and comparing and contrasting them. Writing prepared contrast questions is an excellent way to prepare. And even if the majority of the things you may typically prepare contrast questions on won't actually be prepared contrast questions, but prepared you for thinking deeply about a particular topic and could help you answer a question worded a different way about one of those topics. And then lastly, we do this when we have our groups, when we have our study sessions as a group, but I highly recommend working in groups. So, if you're feeling a little uneasy, perhaps you weren't the most happiest with your last exam grade, I would say study with other people, because other people, finding a study partner, finding a study group, other people pick up on things that you wouldn't necessarily pick up on. And I hope that you've learned that through your in-class group work activities. And it's great that we do work in class, but you also might find that you benefit from working with other people outside of class too. It can be your group, it doesn't have to be your group. It could be your BFF, it could be some random person. You go, hey, want to be my study buddy? Except you won't do that because that's not cool. But several of my friendships were formed by people coming up to me and asking me if I wanted to be their friend. So who knows? Anyway, I always recommend, it's actually true. I have a grand total of 2 friends in Winston-Salem outside of work, and one of them was my friend who works at the TV station asking me if we wanted to be friends at Quilt Guild, and now we're friends, so sometimes it works. Yeah, it sounds really weird in theory, and I was always self-conscious about doing that, but when someone else does it to you, when someone else asks you, it's less creepy than you would think it would be. Anyway, I was going to say, and then, okay, so that pretty much covers it for this. I'm going to give, oh, yes, Caroline. Yeah, of course. Yes, so we did not get quite as far last semester. So there aren't, but what I will say with carbohydrates, I was going to say practice problems with carbohydrates. I will say also, we don't talk about it so much. It's kind of in the syllabus, and you don't see it as much because I don't flash it at the end of a PowerPoint, although I did for carbohydrates. Also, the book problems are a good place to go. The book problems are a good place to go. And also when you're thinking about how to allocate your time, and the types of questions to expect, I'd like to think that the relative distribution of points for questions on the exam, I try to make it close to the magnitude that we emphasize different things in class. So for something we really hammered home, if we covered it in 2 different classes, might want to pay that out a little more attention than something we briefly touched on the end of class. That's not to say that everything that's in the videos or that we covered in class isn't fair game, but you can kind of figure out from class what to spend more time on and what to spend less time on. Okay, and then I was going to give two really quick pieces of unsolicited advice about computers before we get started. So about 2 years ago this time, I was finishing up my dissertation. I went to grad school, and the summer before I went to grad school, some people at the place where I had my summer internship, the Savannah River site, resumed. It sounded a lot cuter when I thought about it in my head. Right. So you guys had a pre-assignment, and the pre-assignment today, it was doable, but it was also kind of a lot. And so I'm going to take a minute and here's the key for the assignment. So you had to most of you labeled the 5' and 3' in class, and you circled the phosphodiester bond. The phosphodiester coming from the fact we have a phosphate group, and it's attached to carbons on either end through an oxygen. That's where the diester comes from. And so we have an A up top at the 5' end, and a C on the 3' end. And you had to draw in the complementary RNA. Now, if you're still confused about how to draw complementary strands, I made a supplementary video on it because apparently I have a thing about supplementary videos. You can go to the supplementary video file. You can look at and I can walk you through how to draw the complementary strand. What I like to do is I generally draw the base first, and you draw the base so it's complementary hydrogen bonding with its partner, because this bond, the glycosidic bond, is rotatable and the base can rotate around. And it will rotate around until a base pairs with something and then it locks. And then I literally just draw the ribose or deoxyribose sugar upside down, and then I connect the 2 bases using a phosphodiester bond. And so because we're going 5 DNA or a DNA-RNA hybrid is a anti-parallel double helix. If we go to 5' to 3' in this direction, we're going to be going 5' to 3', the opposite direction on the base pairing strand. And then 2 characteristics. So you should have drawn in uracil and guanine as the complementary base pairs because this is RNA. RNA doesn't have T, it has U. The difference between T and U, all it is T has a methyl group there, which I'm promptly going to erase because I don't want you to get confused. And then also the other difference between RNA and DNA is that RNA has a 2' hydroxyl that I've circled in pink, and DNA, deoxyribonucleic acid, as compared to ribonucleic acid for RNA, because RNA has ribose, DNA has deoxyribose, comes from that 2' hydroxyl. And when you watch the video on RNA, the structure, you learn that the presence of that 3' hydroxyl makes RNA much more reactive than DNA. It degrades much more easily than DNA. But also it makes it more flexible, and frankly, some DNA chemists might disagree with me, but RNA can do much more interesting things than DNA because of it. So we want DNA to be stable because it is the essence of ourselves. It contains the nucleic information the information that makes us us. But RNA can do a lot of cool other random amazing things too that DNA can't. And it honestly all comes down to, or a lot of it comes down to that presence of a 2' hydroxyl. So chemistry is the shit, y'all, and this is living proof. Okay, but that's a bit of a sidewind. Okay, so what I want you guys to do now is we went over the structure. I want you guys to take a really quick look, and I want you to talk to each other, or actually, no, really quick, what I will say, work through 2 to 4 together as groups, and then we'll check back in. Oh, did I have a different one? Oh, okay. All right, guys. It is the same question. So 5 stacking interactions. List the types of interactions. Add the next nucleic acid, and then try to add ACT and see what happens. And I see. So look at the stacking. Talk about the types of interactions that can happen. Okay. And then try to incorporate that and then try to incorporate it. Talk about the types of interactions that happen. Draw the mechanism of how to add these actions, and then try to add the next nucleic acid. Okay. This is about That's okay too. So the reason I put it there is because I have it with the experience in the parallel, and then the 3' strands parallel. I honestly don't. I was just trying to but I really just want to be able to understand if it starts and stops, perhaps. I'm really more concerned about but that was the guidance the direction of my extending. That's the question I want to ask. I know it's a non-answer, but that's kind of the thing. So, yeah. Okay, all right, so stacking interactions. Can anybody explain to me what stacking interactions are? Would one of my lovely reporters like to tell me about stacking interactions? Yes, Elena. Right, so I have tried in vain to find a definition of stacking interactions that makes sense to me and satisfies my scientific curiosity. I haven't really been able to find that. But the thing that I think of as analogous for stacking interactions is like pi stacking between those bases, because those bases are flat and aromatic, they're planar, they're aromatic, and they just like, for that reason, they just like to nestle on top of each other. So that's like, I just think of the nucleotides snuggling on top of each other. And so if you have two bases hanging out together, the strength of their interaction is dependent on the number of hydrogen bonds. But when you put a bunch of bases together, you also have to think about how they stack and play with each other, because a nucleic acid is this long thing where you have bases stacked on top of each other. Okay, so we know that when we move from thinking about DNA as a bunch of bases together, and we switch to thinking about it, or switching and thinking of DNA bases hanging out versus they all stack on top of each other and they also have a ribose ring and a phosphate backbone, we know that there's hydrogen bonding. We talked about that there's base, but there's some kind of like pi stacking interactions going on. But if we look at the structure of DNA, so here's the model. For some reason, it doesn't have the phosphates drawn in. And here we have a space-filling model of DNA. Have I neglected any other types of interactions that are going to contribute to DNA structure? Has anybody thought of any other interactions that might also be important? Yes, Paige. Right. So another thing to remember that we can't neglect with DNA is when we look at DNA, we have to think of the phosphate backbone. And so those phosphate groups are negatively charged. So the phosphates between the different bases are going to repel each other, and that's also going to help DNA get its characteristic structure. So we have the Watson-Crick bonding, we have pi stacking or base stacking, but we also have those electrostatic repulsions. And the hydrogen bonding, the base stacking, and the electrostatic repulsions are really you could also make an argument probably for the hydrophobic effect too, I would think. But really the big players are going to be those three. And then so for those of you who, the next question I had was,
We talked briefly about, I think, DNA polymerase in the nucleic acid structure video, or perhaps it was the nucleic acids and nucleotides video, where I showed you how nucleic acids are incorporated into a growing DNA strand. And so the way that happens is if we have, so nucleotides, a nucleotide contains a nucleobase, the sugar, the ribose or deoxyribose, and then 3 phosphates. So this would be, oh goodness. So this would be uracil, UTP, so uracil triphosphate. And so the phosphate that's furthest from the nucleobase is the alpha, the beta, and then the gamma. Hopefully I didn't get those confused. We have the alpha, beta, and gamma phosphate. And so what's going to happen is for the next nucleobase to get attacked, the 3' hydroxyl is going to make a nucleophilic attack on the phosphate that's closest to the nucleobase. And we're going to see a chemical reaction that's going to look a lot like when you're adding to a carbonyl carbon, except it's a phosphate. The electron density will go up onto the oxygen. It'll go back down, and then the PPI, or the diphosphate, is going to leave. And later on, we're going to learn why this reaction is favorable. Now, this molecule here, you notice, looks a little bit different than the other nucleobases because instead of a 3' hydroxyl, it has a 3' azido group, or N3. And so what happens when you try to, okay, I drew it in, but what happens when you have a molecule that has an azide instead of an O', or instead of a 3' hydroxyl? Yes, Elena. Right. So what happens is when you have this 3' azide inside of a hydroxyl, it cannot make a nucleophilic attack on the next nucleotide. And so this molecule here is AZT. And so AZT was the first big AIDS drug. So a little bit of history. Back in the day, in the early 80s, people started getting AIDS, and people were dying because AIDS severely suppresses your immune system response, and so it makes you more prone to being sick and dying from things that people with normal health would be able to weather fairly easily. And so it was really scary. People didn't know how it was transmitted for a while. And there wasn't a cure for the longest time. So my aunt actually is an infectious disease doctor. And when she started working back in Florida, just an hour or two north of Miami back in the late 80s, it was basically still a death sentence. And now they're actually like curing AIDS and having people who are having children that are HIV negative. So there's been such strides that have been made. But one of the first things to be prescribed was AZT. And the way this works is viruses would incorporate this into their genome, and then they wouldn't be able to continue replicating their genetic information, and so it would kill the virus. But AZT was hard to take because it had really crappy side effects. So why would this be a problem? Why would this cause bad side effects for someone taking this drug? So we know it's going to kill the virus, but what else is going to, or I guess you can't really kill a virus, it's going to stop the virus from replicating, but how else would this be a problem for like just a normal human? Yes, Greg? Exactly. So it's really hard to selectively give this to a person so it only hurts the virus. So it's going to get incorporated potentially into your own replication of your own genetic material. And so cells that replicate a lot, like your hair and your nails, are going to be targeted as well. And so this can cause really nasty side effects. And this is the same principle for a lot of cancer drugs that target cells that rapidly divide. So that's why, along with targeting cancer cells, you also oftentimes lose your hair with chemotherapy. So just a side note into medicine world there. And then one last question. I wanted you guys, I'm not going to have you report back to the class, but I want you to think in your group and decide about if you have an A or a T or a C and a G within the context of a DNA strand, which is going to be stronger and why would it be stronger? So make a decision, about which you think will be stronger and why. And with that in the back of your mind, we're going to take out our Foundations of Biochemistry book. It's back. And I'm going to have you work. We're going to start working on, so we'll go to page 86. We're going to go to page 86. And you're going to work on 2 to 5. And we can do backward. We got several markers up here. I'm going to move my computer so you have plenty of room.
Okay, perfect.
Yep.
If we're seeing the bases are more
Yeah, we can put these. We have lots of options. All the options. Okay.
Yes, it was. Okay. So this is just submitted, the creating that's submitted by email, that's submitted later before I print it. So I will email you this. You may have to remind me, I will write it on my hand. I need to see. But I have it, it's great, have a great. Yes. I also realized, but I forgot to send in our activity from Thursday as
And I'm just literally copying your answers. So those of you, it was like take a minute, look at the answers on the board, and see if you agree with the answers that the other groups came up with, and if not, we can come back up to the board and make some changes. So while we're waiting, make sure you agree with what's on the board, check what you have, see if there's any discrepancies. Okay. All right, so we'll take a minute. Okay, so in the book. All right, so which is more hydrophobic, the sugar phosphate backbone or the nitrogenous base pair? Yes, I would say the nitrogenous base pair is more hydrophobic because it has that flat aromatic ring. It has heteroatoms on it, but it's also a big ring that has double bonds and hydrogen's coming off it. It's probably going to be it's more hydrophobic than the sugar phosphate backbone, which has negative charges. We know water loves to solvate negative charges. And if you were to predict which of these two components would be excluded from the aqueous environment, well, the more hydrophobic one, which would be the nitrogenous base. And if we look at the structure of DNA, we can see here that the bases are contained in the center. And although it's represented simplistically as a ribbon here for visual purposes, the sugar phosphate backbone is what's hanging out and interacting with the water and making sure our DNA doesn't crash out of solution. Okay? We saw when we talked about carbohydrates what happens when you don't hydrogen bond with the water solution. There's not necessarily a problem, but it's something you don't want to happen if you want something to be soluble. Okay, so next, all right, single-stranded RNA also forms a helical structure. Okay, so when we think about DNA, we automatically think about Watson-Crick base pairing. But Watson-Crick base pairing can't be the only thing that holds DNA together or RNA together because you have single-stranded nucleic acids. And so like you see here, we have a picture of single-stranded RNA. So we have to start thinking about the other interactions that can hold the RNA together. And so, yes, there's hydrophobic interactions going on here. And there's also hydrogen bond. There can also be hydrogen bonding going on between groups. But some other interactions that we could think about too is we also have the electrostatic repulsions between the phosphate backbone. And there's also a new type of interaction that we talked about in the pre-portion of the class. So how are we going to ensure that those bases are lining up? What types of interactions have to be going on for that to happen? The stacking interactions. So the stacking interactions are huge for making sure those bases line up, even when there isn't a Watson-Crick partner across the way from it. Yes, sir. So you have had, so I guess if you have the bases somewhat exposed to water, in theory, some of those
bases could be hydrogen bonding with each other. They could be hydrogen bonding with water because you do have double bonded oxygens. You have like nitrogens hanging off. They're hydrophobic parts. You could also be solvating the hydrophilic parts as well. So probably the base stacking is going to be really big right now. The base stacking, the hydrophobic effect, the electrostatic propulsion, so the backbone, it's all of those things together. But we have to think about the base stacking in terms of what lines up the bases as well. And the point of this exercise is for you guys to see that base stacking matters. And then if you went through, we identified the non-covalent interactions that occur between complementary. Okay, complementary, we see the H bonding, so specifically the Watson-Crick. And then the interactions that occur between consecutive base pairs. So what causes those bases to line up are the stacking interactions. And then lastly, explain how the orientation of the bases in models 1 and 2 contribute to the strength of interactions between consecutive base pairs. Well, the consecutive base pairs are close together in space, which increases the strength of the pi stacking interactions. So we see that the fact that the bases are close together increases the strength of the interaction, but also the Watson-Crick base pairing makes sure that you have the bases in the correct order as well. Okay, so we're going to take a minute now. Before we move on to the next exercise, we're really going to dive into the relative importance of hydrogen bonding and pi stacking. We need to take a minute and talk a little bit about DNA structure. So I'm going to do a little bit of lecturing here on some of the material that we didn't cover in the video. And that's the idea of DNA melting. Okay, so if you were to take a solution of DNA and put it into a UV visible spectrophotometer, what you would find is that nucleic acids absorb in the UV with a lambda max of around 260 nanometers. And if you see in this picture here of a generic absorbance plot, you see that maximum. I'm going to fail at drawing a straight line. You see that maximum of 260 nanometers. And that's a reliable way for using Peir's law, so alpha equals epsilon BC, to quantify the concentration of nucleic acids in a solution. It's awesome. I love it. One thing you'll notice if you look closely at this plot is the fact that single-stranded DNA absorbs more strongly than double-stranded DNA. Why is that? So, one of the reasons why this happens, so single-stranded DNA absorbs more strongly than double-stranded DNA. Whoops. It's because, oh, and that was miserable there. It's because the base stacking, the extra base stacking that you see with double-stranded DNA decreases the absorbance at 260 nanometers. All right, so the chemical and structural difference between single-stranded DNA and double-stranded DNA leads it to have different chemical properties. And we can see that when we look at its absorbance. And you have to take that into account when you're quantifying DNA. Okay, so one of the ways we could talk about like the stability of DNA is to talk about its melting temperature. And so the melting temperature is the energy needed, refers to, yes, Sydney?
Wait, but don't they both?
Yes, but because you have more stacking per molecule if you have a double-stranded molecule. Yes. Okay, good question. So the energy needed to take double-stranded DNA and to turn it into two pieces of single-stranded DNA, we refer to this as melting. And we think about it specifically, we could call it melting because you have to apply heat. So you have to put energy to the system in terms of heat. It doesn't necessarily have to be heat, but heat is generally what we use in the lab because it's easy to heat up a tube of DNA to make it go from double-stranded to single-stranded. Now, why do we care about melting DNA? Well, out in the wild, we melt DNA for transcription, for replication, which is important for gene expression. So for the polymerase and other proteins to get in and do their work, you have to pry the DNA apart.
And then practically speaking, we can think of, for those of you who are definitionally minded, we can think of this in terms of being an indicator of the stability of the nucleic acid. So the melting temperature is the temperature at which DNA is 50% single-stranded and 50% double-stranded. So if you have a higher TM, you need a higher temperature to denature. So you need to put more energy to break up the DNA. And this indicates that it's more stable and it has stronger interactions internally. So if you higher TM, takes more energy to break it apart, it's going to be a more strongly held together piece of DNA because you need to put in more energy in terms of heat to break it apart. So experimentally, how does this work? So we can monitor the transition from double-stranded DNA to single-stranded DNA by a decrease in absorbance at 260 nanometers. And so what we can do is we can increase the temperature. We can observe the absorbance at 260 nanometers. And as the percentage of single-stranded DNA goes up, the A260 is going to go up. And what we're going to see is a sigmoidal curve. Oops. So if these are two DNAs with different melting temperatures, we're going to see a sigmoidal curve. And that's because we can think of DNA being like a zipper. So if you pry open one base, it gets easier for the next base to be pried open as well. And so in your groups, as a transition into our group work section, I want you to look at these two DNAs, literally take a minute, and I want you to determine which of those two DNAs, the pink DNA or the blue DNA, is more strongly held together and would be more stable.
I like the answer to this. Oh, you can't see because I'm sorry, you guys are not black and white. Because my math is being fixed by the math people in the factory, I have to print my work computer and it defaults to black and white. Sorry, guys. I know you're pampered with your colored printouts. Okay, this is the point where if we had clicker software, I would do this by clicker so you wouldn't feel intimidated. But it's okay because we know that getting things wrong is a big part of the learning process. And for every time something went right for me in the lab, I had 10 things go wrong. It's a part of life. So that being said, how many of you think the blue DNA is more stable? Okay. How many of you think the pink DNA is more stable? Yes. So the pink DNA in this case is more stable because it has the higher melting temperature. So it has a melting temperature around 85 versus around 82.5 for the blue DNA. And so it'll be more stable because it's going to take more energy. You have to crank it up to 85, an extra 2 to 3 degrees to get it to be half double-stranded, half single-stranded. All right. So now that we talked about DNA melting, we're going to do an activity. We're going to literally look through different sequences, and you're going to parse apart what makes DNA more or less stable. So we're going to work through 6C, 6B, 6C, 6E, and 6F. Okay? So I want you to fill out, to complete these tables, work through these exercises, and then we'll discuss back as a class. And this time I'm actually going to have you fill in the answers on my computer, the reporters, okay? So don't worry about me. Don't be intimidated by the computer.
Go ahead and get to work, and I'll filter through. You guys get all like scared. Okay. We're good. We're fine. We're totally fine. It's all fine. Okay. All right. And then you can click on that, and then there's the eraser if you need it. You're intelligent, you can figure it out. That'll also be able to be helpful.
Yes. Yes. Electrostatic and charge are two different ways of referring to the same interaction. So interactions between like or opposite charge.
How do you even write it?
I don't know. Does she have like a thing? I don't know where it is.
I use my
Use your finger.
I feel like that's gonna be a disaster.
Is there? How else?
Is there
Do you have a pen? Do you
write?
use a pen to write on, or like a
things
stylus?
or do you use your
Yeah,
fingers?
so this is, it should go on the side of the computer. Left, right side. You'll see it. Watch out.
Oh, it's in the computer.
Because it likes to fall out in the bag. Scare the crap out of me.
I was like looking for it on the table. I was
Yeah, I was
like,
like, where?
this is like weird. I'm just gonna.
Yeah, that's good. If you scroll. Oh gosh. I do too. Oh my gosh, this is hard.
I can give you that. I'll end up giving you one.
Oh my gosh, this is really hard.
You're gonna get more of the work with it.
Oh my gosh.
I've seen you write up
It's a struggle. I don't know how she does either. Oh, sequence.
awkward angles. Like I definitely could not.
Oh my god, it's such an awkward one.
I feel like
How do I scroll?
what?
How do I scroll?
I have no idea. I don't know. I want to like touch it, but I don't.
How do I scroll up on
Okay,
this?
okay.
How do I scroll up?
Oh.
Oh. Yeah.
Where we're at.
Yeah, it's a touch screen, which is
Oh,
super
cool.
nice.
I'll pull out my paper.
So like this was only pushed to like that. We'll look at the page there. And we're saying that with A to B, the A was security. So we would also it would be the hash code. So
Yeah, no, you're right. Honestly, it gets everybody.
Therefore, we need a higher.
Oh, wait, shoot.
All right, let's get these back to the front of the class for a minute. Okay. All right, so if we look at, okay, table A, what table A is doing, you have 3 sets of DNA strands. So the difference between the first strand and the second strand, as I've been looking in on your notes, you wisely assessed that the difference between 1 and 2 is the number of base pairs. Now, the composition doesn't really change much because it's all A's and all T's. And so what you see is when you go from 18 hydrogen bonds, or 9 base pairs, 10 base pairs, or you increase the number of hydrogen bonds, you're seeing an increase in the melting temperature. Then when you go from number 2 to number 3, you have the same number, or you have a fewer number of base pairs. You have the same number of hydrogen bonds. You see that the stability still goes up, even though you have fewer bases and the same number of hydrogen bonds. Okay, so it looks here like, so the number of hydrogen bonds does matter when you're going from, it looks like the hydrogen bonds could matter when, especially when you're going from the first DNA strand to the second DNA strand, but that's not necessarily the case. Because if the number of hydrogen bonds were only what mattered, then when you go from this DNA, the number 2 to number 3, in theory, you would say the same melting temperature, right? If it was only hydrogen bonds that mattered. So at first glance, and even I have in my notes, I do have that, yes, it does appear to scale with hydrogen bonds. But if you were working on this and writing the answer, if you felt a little bit uneasy, like maybe it was a little more complicated than that, your brain was absolutely working in the right direction. Okay? And so if we move on to the second set, and if we're looking here, if we're comparing all the DNAs together, okay, we see they're all the same number of base pairs. So things were a little bit weird here because you were changing the number of bases, you were changing the number of hydrogen bonds, and for this one, adding an extra base mattered, but for this one, if you took away a base, it was still stronger. So it's kind of weird. So with this one, we're removing away one variable, and we're saying, okay, all of these are the same length, but they have slightly different amounts of hydrogen bonds. So when we look at these two, we can see that when we switch from A and T to being T and A, and then in this one, we switch C and G from being G and C, we can see that between the first 2 DNAs and the second 2 DNAs, our number of hydrogen bonds are changing. And generally speaking, when you shift between this 2 set and that 2 sets, so when you're going from switching out an A and a T to a C and a G, you're seeing that the melting temperature is increasing. So when you're switching, so when you're adding an extra hydrogen bond, you're seeing the temperature increase. So the number of hydrogen bonds in theory could matter. But something is wonky is going on. And you guys picked up on that in 6F. That even within the same DNA sequence, you're just switching on what side the C and the G are, or what side the A and the T is. You're getting a significantly different melting temperature just from flipping the base pair around. And you would think if only length and hydrogen bonds mattered, you would get the exact same melting temperature. But when you switch around which side the C and the G are on, or the A and the T, you get a different melting temperature. Yes, Sydney?
Does that have to do?
Exactly.
So what happens is that we're not going to get into the details of the hierarchy, or maybe a little bit, actually. But what this is telling us is that the order that the bases are in can be either better or worse at stacking. And so that stacking matters. So hydrogen bonds and length matter. And if anybody who's designed a primer for research lab knows that length and composition matter. But the thing that puts it over the top is how well does it base stack? And so certain bases are going to stack better than others. And so if we see here, if we look at these melting temperatures, it looks like which of these oligomers has the highest melting temperature? Number 3. And if we look at number 3, C stacking with A appears to have the strongest base stacking. Okay, what's the second best? Or I guess it with A and C and then T and G. What's the next one? Yeah, number 4. So what's going to be next on the hierarchy is A with G and T with C. Again, it's a little counterintuitive because you would think maybe that purines would play better with each other at pyrimidines, but that's not necessarily the case. Okay, what's the third highest? First one, yes. So A stacking with itself and T stacking with itself is next. And then what's last? Alternating A's and T's. So I don't know exactly why some of these base pairs play better with others. I imagine you could do some fancy PChem experiment where you map the electron density and figure it out. I'm not so worried about that. What I'm worried, or what I'm concerned about, is that you guys understand that, first of all, generally, by throwing a C and a G in there, it's going to be better at, the C and G's generally are better at better at base stacking than A's and T's alone. But the neighbors that are above and below our particular base pair are going to influence the stability by how well they stack on top of each other. Yes, Maddie? So I don't know where they would fall. And I'm not so concerned about you being able to make an exhaustive list, more so that C's and G's generally stack better and the order matters. That's what I would say. And so I would say know this hierarchy, and I'm not so worried about how C's and G's would fit in there. Yes, Sydney? Yes.
So then, you were looking at it, would you say that the stacking is more of an influence or the number?
I would say that these data show that the stacking is really what matters.
Okay, so even though like the 40.3 that's more because of the stacking because
That's tough. You know what? I would say that stacking and hydrogen bonding matter, and we can't neglect either. I think that's fair, understanding that both are important and both come into play. It's really easy, and oftentimes we fall into the trap of assuming it's just Gorensek base pairing, and it's not. And that's, I think, what this exercise is designed to illustrate. Okay, now, before we move on, we're going to see why the sequence of DNA, so we understand that the sequence of DNA matters in nature because DNA codes for RNA, which RNA can float off and do all kinds of cool things. But canonically, mRNA goes to the ribosome and it's translated into a protein. We know that. We've hit on that many times. But thinking about the sequence of DNA matters because we've seen that the sequence of DNA determines how well it can be melted, but also it determines how well the DNA can be melted determines how well it can be manipulated. And we care about manipulating DNA in the cell. So if we think about, if we do a quick foray into gene expression, if we're hanging out in the genome of an organism, We have a gene, which is a fancy name for DNA that codes for RNA that codes for protein. Now, there's a short sequence of DNA that comes before the gene, which is called the promoter. But before and the promoter is where the polymerase comes in and makes a copy, so if it's RNA polymerase, it'll go in and make the mRNA, which then goes to the ribosome, which then gets turned into protein. And you've probably heard of promoters perhaps in your introductory biology class or if you've taken genetics. But oftentimes there is a sequence before the promoter that clues in the polymerase that, hey, there's a promoter here, and there are other accessory proteins that can anchor onto that spot and then pull in the polymerase to get it to sit on the promoter. And one of these sequences, and this is found in eukaryotes and in archaea, it's called the Tata box. And the sequence for the Tata box is as such. And what happens is this protein called the Tata box binding protein, or the TBP, comes in and it sits on the Tata box, and then it pulls in RNA polymerase and gets RNA polymerase to sit on the promoter, and then it transcribes the gene into RNA, which is then translated into protein. And again, remember that the box is, in this case, is signaling double-stranded DNA. And so the next couple of exercises, we're going to talk about how the sequence of DNA can determine how things, can come in and bind the DNA and then have it translated into RNA. So I'll let you guys work on problems 2 and 3, all right? And that's how we'll close class. Okay. I'm actually going to put the pictures up so you can see number 2.
Specifically for number 2, I'm having you think about which groove of the DNA is distomycin binding to. Grooves. Maybe I should throw up the PowerPoint here.
We have the major groove and the minor groove of DNA. Does anybody remember which of these is the minor groove? Yes? The minor groove, I see some motions like this. So the minor groove is the smaller portion. So it's going to be the second groove down here. And then the major groove is the wider portion where the phosphate backbone is further apart from each other. So that being said, you can use that to help you identify where distomycin is binding. Pardon? Yes, that's why I put it up on the board. But I wasn't going to reprint 20. I know.
So there was some confusion about the video where it showed an illustration of, if you look at a Watson crick base pair, of which part's the minor groove. The minor groove and the major groove, when I showed that illustration, it was just referring to which part of the base pair
is exposed in the major and the minor groove. So if we look at this PowerPoint here, what we would think of as the top of the base pair, like that top edge, would be exposed in the major groove. And what we would think of as the bottom part of the base pair, it would be exposed in the minor groove. So it's not that certain bases are exposed, or that the minor groove only occurs on certain bases. It's just that it's a matter of which part is exposed. And I had two base pairs here, but the minor groove would be on the bottom of this base pair, and the major groove would be exposed on the top of this base pair as well. Okay, all right, carry on.
So the minor groove is generally the skinnier. Okay, so if you look at that structure, the green is the distomycin. The minor groove is the skinnier portion of it, the skinnier lower grooves. Okay, so if you look at the structure, the distomycin is the green, the minor groove is the smaller. Confused.
Yeah. Okay, all right, we'll come back, okay. All right, so unfortunately, we are running out of time, which is very sad, but, okay, so what I will say, the point of this exercise is, so the molecule I showed you here is called distomycin. Distomycin is a class of like little crescent-shaped molecules that bind to the minor groove of DNA. And so they're crescent-shaped, and so they just slide right in. And so the point of this exercise, what I was meaning to have you guys do is if you look at the picture from the PowerPoint pre-class video, we have the major groove and the minor groove. And you see the minor groove's a little bit skinnier than the major groove. It's hard to find a picture, it's hard to find a crystal structure where you have a long enough piece of DNA that you're able to be a good job of being able to see the major and the minor groove, and which is one of the reasons why this picture wasn't the greatest. But this would be the minor groove because the backbones are closer together than out here, which would be the major groove. So this is distomycin binding to the minor groove of DNA. And so these small molecules, and you can change the identity of the rings that are in them to have them bind to different sequences of DNA. And that's actually an active area of research, is developing small molecule drugs that bind to specific sequences of DNA through the minor groove. And so what what I'll do is I'll have you guys, I will bump number 3. I'll bump number 3 to homework. I'll make a video about it, and I'll look at it when you come back from spring break. But yeah, because what this is doing is this is talking about how the composition of the DNA sequence can determine how well different things can bind, and that has a magnitude on gene expression. Okay, so we've hit 10:30, so I will cut you guys loose. I will